Welcome to Frontier Flash. I'm going to keep this precise and evidence grounded. Because longevity science is an area where the signal and the noise are genuinely difficult to separate. Today, we're looking at 5 habits that have the most consistent support across current research, not fringe theories, not supplement stacks, but behaviors with meaningful biological plausibility and replicated findings. The first is Zone 2 cardiovascular training. This refers to sustained aerobic effort at a relatively low intensity, the kind where you can still hold a conversation, but only just. Research into mitochondrial function suggests that this intensity range is particularly effective at increasing mitochondrial density and improving metabolic flexibility. Several large observational studies tracking cardiovascular fitness as a predictor of all-cause mortality have found that cardiorespiratory fitness may be one of the strongest modifiable predictors of lifespan we currently have access to. The mechanism isn't fully resolved, but the consistency of the finding across different populations is notable. The second habit is time-restricted eating, sometimes called intermittent fasting, though that term covers a wide range of protocols. The version with the most research attention is a compressed eating window, typically somewhere between 8 and 10 hours, aligned with daylight hours where possible. The proposed mechanisms involve circadian rhythm alignment, improvements in insulin sensitivity, and the upregulation of cellular repair processes, including autophagy. It's worth stating clearly that most human evidence is still observational or short-term. Animal model data is more robust, but translating that directly to humans requires caution. Third, resistance training, specifically in the context of preserving lean muscle mass as we age. The process of muscle loss with age, sarcopenia, is associated with increased mortality risk, reduced metabolic rate, and declining functional independence. Resistance training is currently the most reliable intervention we have to slow or partially reverse this process. Some longitudinal data suggest that maintaining or building muscle mass in midlife may have significant downstream effects on health outcomes in later decades. The dose required is lower than many assume. Even 2 sessions per week appears to have measurable benefit. The fourth habit is sleep duration and quality. This one is easy to understate because it lacks the novelty of newer interventions, but the research base is substantial. Consistently sleeping fewer than 6 hours per night is associated in multiple large studies with elevated risk across a wide range of conditions: cardiovascular, metabolic, and cognitive. Mechanistically, sleep is when key biological maintenance processes occur: synaptic pruning, metabolic waste clearance via the glymphatic system, and hormonal regulation. Crucially, chronic sleep restriction appears to compound over time in ways that are not fully reversed by short-term recovery. Fifth, and perhaps the most underappreciated in mainstream longevity discussions, social connection. This is not a soft finding. Large meta-analyses examining social isolation and loneliness have found associations with elevated mortality risk of a magnitude that is comparable to, and in some analyses exceeds, that of more commonly cited risk factors. The biological pathways under investigation include chronic inflammatory signaling, HPA axis dysregulation, and downstream effects on immune function. The effect appears to be dose-dependent. Quality of connection matters as well as frequency. Taking these 5 together, what's worth noting is that they are not independent interventions. They interact. Better sleep supports training recovery. Social engagement is associated with better adherence to health behaviors. Time-restricted eating can improve sleep quality in some individuals. The emerging picture in longevity research is less about isolated variables and more about the integrated system, a cluster of behaviors that reinforce one another across multiple biological axes. Now the caveats, and they matter. Longevity research faces serious methodological constraints. Most human data is observational, which means we are working with associations, not confirmed causation. Confounders, socioeconomic status, baseline health, genetics, are difficult to fully control for. The populations studied are frequently not representative, and the timescales involved make randomized controlled trials extremely difficult to conduct. None of this invalidates the findings, but it does mean that confidence levels vary considerably across the evidence base. Looking forward, the most interesting signal in this space may be the shift toward personalization. Research into biological age clocks, epigenetic markers that attempt to measure physiological aging independently of chronological age, is gaining traction. If these tools become reliable and accessible, they could allow individuals to measure their actual response to behavioral interventions, rather than relying solely on population-level averages. That's a meaningful shift in how we might approach this science. That's the briefing. Frontier Flash covers the research frontier, not the headlines. If you found this useful, the subscribe button keeps the signal coming. See you in the next one.